Anyone can tell what we have discussed in last class? In second minute, what are the topics we have covered till now? இதுவரைக்கும் என்ன பார்த்துருக்கோம் செகண்ட் யூனிட்ல? What are the topics we have covered? Unmute and tell. Reshma. Neha. Priya.
Sir.
லாஸ்ட் கிளாஸ்ல என்ன பார்த்துருக்கோம் இது வரைக்கும்? லாஸ்ட் கிளாஸ் வந்தீங்கல்ல?
எஸ் சார் வந்தேன்.
என்ன பார்த்தோம்?
சரியா ஞாபகம் இல்லை சார்.
ஞாபகம் இருக்காது. நம்ம நோட்ஸ் எடுத்தால் தானே ஞாபகம் இருக்கும். அகிலா?
எஸ் சார்.
லாஸ்ட் கிளாஸ்ல என்னவா பார்த்தோம்?
Containment level, sir.
Okay, then Sashri Arsha. Shruti. Akshata. Riyas.
Sir.
Last class la enna discuss பண்ணோம்?
லாஸ்ட் கிளாஸ் சார், லாஸ்ட் கிளாஸ்ல நீங்க நோட் முடிக்கலன்னு திட்டினீங்களா? அதான் நோட் எழுதுறேன்னா இதை கவனிக்கணும்.
ஆஃப்டர் தட் அதுக்கப்புறம் கொஞ்சம் டிஸ்கஸ் பண்ணோம் இல்லையா? என்ன டிஸ்கஸ் பண்ணோம் கடைசியா?
அதான் சார் கண்டெய்ன்மெண்ட் பார்த்தோம், அண்ட் தென் வேறு எதுவும் பார்த்தோம் சார்.
We are in a recorded meeting only. Last class we have discussed about biosafety cabinets. Biosafety.
Yes sir, ஞாபகம் வந்துச்சு.
Cabinet. So in the second unit we have discussed about biosafety and biohazards. So biosafety levels, what are the biosafety levels? 4 stages are there. And then we have discussed about the containment levels, primary containment, secondary containment. Then we have discussed about the what is the bio safety cabinet, what are the levels are there, what are the 3 stages are there in the biosafety cabinet. And then we have discussed about the pathogens and what are the risk groups available. 4 risk groups are there associated with the pathogen while we are working in the laboratory. So these are comes under the biosafety and biohazards, which on which we are working in the research laboratory or in a particular organization. So in this class we are going to discuss about good laboratory practice, which is also to be followed in the research organization or the research institute wherever we are working under some research projects. So good laboratory practice is called as the GLP. It is an FDA regulation. FDA is nothing but Food and Drug Administration. So what is the definition? GLP embodies a set of principles that provides a framework within which laboratory studies are planned, performed, monitored, reported and archived. So it is said to be good laboratory practice. So we have to framework. We have to do a chart or a framework in which the laboratory has to be carried out. The laboratory research has to be carried out. We have to plan our work, we have to perform the experiments, and then we have to monitor the experiments, and then we have to report it, and then we have to finally consolidate the report, and we have to archive the data for the future use. So this is your definition for good laboratory practice. So what is the history behind this good laboratory practice? It is a formal regulation that was created by the Food and Drug Administration, that is United States Food and Drug Administration in 1978. Although GLP originated in the United States, it had a worldwide impact. Even though it was created in America, it has worldwide impact, good laboratory practice. Non-US companies that wanted to do business with the United States or register their pharmacies in the United States had to comply with the United States GLP regulations. So non-US companies, இப்போ இந்தியாவில் இருக்கிற நிறைய companies, they are working for US based pharmaceutical pharmacy drugs, pharmaceutical drugs they are producing. So in which they have to compile the in which they have to follow the rules and regulations according to the United States GLP regulation. So India company irundalum, if even though the company was in India, if they are working for US based company, US based products or US based drugs, they have to compile the they have to follow the rules and regulations of GLP according to United States. So this is the protocol. They eventually started making GLP regulations in their home countries also. So அதை பொறுத்து என்ன பண்ணாங்க, several countries in India or some other countries also, they had framed some certain regulations according to the GLP. அவங்க individual companies வந்து they have framed a certain set of rules and regulations. In 1981, an organization named OECD, Organization for Economic Cooperation and Development, produced GLP principles that are international standard. So in 1981, ஒரு ஓவராலாக ஃப்ரேம் பண்ணாங்க ஒரு ஆர்கனைசேஷன் ஃபார் எக்கனாமிக் கோஆபரேஷன் அண்ட் டெவலப்மெண்ட். தே ஹேட் ப்ரொடியூஸ் தி ஜிஎல்பி பிரின்சிபல்ஸ் தட் ஆர் இன்டர்நேஷனலி ஸ்டாண்டர்ட். ஸோ எல்லாருக்குமே காமனான ஒரு பிரின்சிபல் மாதிரி தே ஹேவ் ஃப்ரேம்ட் இட். So why was this good laboratory practice was created? In the early 70s, FDA became aware of cases of poor laboratory practice all over the United States. ஸோ அமெரிக்காவை சுற்றி உள்ள எல்லா லெபார்ட்டரியும் பார்த்தீங்கன்னா ஒரு கைட்லைன்ஸே இல்லாமல் ஒரு 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 குட் லெபார்ட்டரிக்கான எந்த ஒரு எஃபிஷியன்ட்டுமே இல்லாமல் அங்கே நிறைய ரிசர்ச் போனதை தேவ் ஐடென்டிஃபைட். FDA decided to do an in-depth investigation on 40 toxicology labs. They discovered a lot of fraudulent activities and a lot of poor lab practices. So அவங்க ஐடென்டிஃபை பண்ணதில் நிறைய ஃப்ராட் ஆக்டிவிட்டீஸ் நடந்துச்சு. Poor lab practices have been followed in the toxicology laboratories. Examples of some of these poor lab practices found were, என்னென்ன எக்ஸாம்பிள்ஸ்லாம் இருந்ததுன்னா, equipment not being calibrated to the standard form, therefore giving wrong measurements. ஸோ என்னென்ன பேராமீட்டர்ஸ்லாம் தே ஹவ் ஐடென்டிஃபைட் அஸ் எக்யூப்மெண்ட் நான் வந்து கேலிப்ரேட் பண்ணியிருக்க மாட்டாங்க. ஸோ அதனால் என்ன ஆகும், நமக்கு ராங் மெஷர்மெண்ட்ஸ் வரும். ஸோ தர் இஸ் அ protocol where our instruments have to be calibrated on a yearly basis or in a certain duration. So the each instrument making, they will be giving a protocol when needs to be calibrated at time interval according to the nature of the equipment. They have to calibrate it. Inaccurate accounts of the actual lab study, so those were also found. An inadequate test system, so there is no adequate test system followed in the laboratories. So these are some of the poor lab practices they are found out from the toxicology laboratories. One of the labs that went under such an investigation was came in headline news. Name of the lab was Industrial Bioethics. This was a big lab that ran tests for the big companies such as Procter and Gamble. So it was discovered that mice that they had used to test cosmetics such as lotion and deodorants had developed cancer and later they died. So அவங்க வந்து ஒரு லோஷன் ப்ரிப்பேர் பண்ணாங்க ஃபார் தி காஸ்மெட்டிக்ஸ்க்கு. ஸோ அது லேட்டர் ஆன் என்ன பண்ணுச்சு, இட் ஹாஸ் கிரியேட்டட் கேன்சர் இன் தி ரேட் அண்ட் இட் வாஸ் டைட். ஸோ இண்டஸ்ட்ரியல் பயோடெக் லேப் த்ரூ தி டெட் மைஸ் அண்ட் கவர்ட் ரிசல்ட்ஸ் deeming the products good for human consumption. So அதே என்ன பண்ணாங்க, அதை மூடி மறைச்சிட்டு they had said it is good for human consumption, lotions or deodorants in the cosmetic field. So those involved in production, distribution and sales for the lab eventually severed jail time. So இது வந்து later on கண்டுபிடிச்சதுக்கு அப்புறம் they have been punished. So this was the main example for this poor laboratory practices. So what is the objective of GLP? What is the main objective? It makes sure that the data submitted are a true reflection of the results that are obtained during the study. So அது என்ன பண்ணும், GLP அது வந்து அஷ்யூர் பண்ணணும். They have to assure the data submitted அது வந்து அவங்க பண்ண ரிசல்ட்லேருந்து வந்த டேட்டா தானா அப்படிங்கிறது they have to assure it. GLP also makes sure that the data is traceable. So we can trace the data. அது வந்து ஈஸியா நம்ம ட்ராக் பண்ணி பார்க்க முடியுமா? We can GLP has to assure that the data is traceable. அது வந்து நம்ம ட்ராக் பண்ணி பார்த்தா the same data should come. So அந்த ரிசல்ட்ஸ் வந்து அதே மாதிரி இருக்கணும். It promotes international acceptance of test. So the GLP நம்ம ஃபாலோ பண்றது மூலமா it will provides or promotes the international acceptance for the test. So what is mission of GLP? Test system should be framed, and then archiving of records and materials should be proper. Apparatus, materials and reagents facility must be provided to the research laboratory. Quality assurance programs must be framed in every institution or in the research organization. Performance of the study has to be performed, and then reporting of the study results must be documented, and standard operating procedure for each and every equipment must be framed, and then personnel and test facility organization must be there. Personnel, the equipped personnel must be there. He must be well qualified with the standard protocol, and test facility must be there in the organization. So these are the main missions of the good laboratory practices. So what is standard operating procedure? SOP is nothing but standard operating procedures, written procedure for a laboratories program. So it may be for a particular laboratory or for a particular equipment. They define how to carry out protocol specified activities. This standard operating procedure will define how to carry out the protocol specified activities. Most often written in a chronological listing of action step. So step by step நம்ம எப்படி follow பண்ணனும் அப்படிங்கறத it will be mentioned in the SOP. They are written to explain how the procedures are supposed to work. So on the SOP தான் how the procedure is going to be performed for each and every experiment or the equipment handling. அதெல்லாம் வந்து they will explain. So for SOP, what are the things are needed? Routine inspection must be done whether this SOP is followed. Cleaning, maintenance, testing and calibration are to be performed in the equipment wise. Action to be taken in response to equipment failure. Equipment failure ஆயிடுச்சுன்னா we have to call the vendor and then we have to rectify it, and then the equipment has to be on process. It has to be ready for the research. Analytical methods, we have to search for the different analytical methods for the experiment. And then definition of raw data, what are the raw data available, we have to mention it. Keeping records, reporting, storage, mixing and retrieval of data must be there. So each and every experiment performed must be recorded and has to be retrieved whenever it is needed. So what is the statistical procedures for data evaluation? So என்ன statistical procedure follow பண்றாங்க இந்த data evaluate பண்றதுக்கு? Statistical procedures are not simply chosen from a textbook. அது வந்து ஒரு textbookல இருந்து எடுக்கிற ஒரு data கிடையாது. Statistical dataங்கிறது. It is a practitioners in a particular field may adopt certain standards which are deemed acceptable within that field. So if one is familiar with the molecular docking, he has to frame a certain standard regulations. These are the experiments how to be followed with the certain guidelines. So this has to be acceptable within that field, and the field of the persons are that protocol has to be accepted. So regulatory agencies often describe acceptable statistical procedure. So on the regulatory agencies என்ன பண்ணுவாங்க, எதை வந்து நாமினலா அக்செப்ட் பண்ண முடியுமோ அந்த ப்ரொசீஜர் தான் they will be following. ஓகேவா? ஸோ எல்லாராலையும் அக்செப்ட் பண்ணிக்கக்கூடிய ஒரு SOP ப்ரொசீஜர் தான் they will be following. So அந்த ப்ரொசீஜர் எதுலேருந்து எடுப்பாங்க? It has to be obtained from the statistical data. So instrument validation. This is a process necessary for any analytical laboratory. So instrument வந்து validate பண்ணி வச்சுக்கணும். Data produced by faulty instruments may give the appearance of valid data. அது வந்து நமக்கு ஒரு valid data கொடுக்குற மாதிரி இருக்கலாம், but அது வந்து நமக்கு வந்து accurate data கிடையாது. The frequency for calibration, revalidation and testing depends on the instrument and extent of its use in the laboratory. As said before, so the frequency for calibration, revalidation and testing is beyond. It depends on the instrument and how we are using in the laboratory. often regular use பண்றோமா or we are using it on what in a temporary basis. If you are using for a month, if you are using 1 or 2 times, or if you are a spectrophotometer you will be using daily, it will be in a frequent use. So according to the instrument depends, we have to calibration and validation has to be performed. So whenever an instrument's performance is outside the control limits, reports must be discontinued. So ella protocol for every experiment you are performing, there is a control limit. So the control limit தாண்டி போச்சுனா we should not consider the data. Okay, so it must be discontinued. The instrument performance வந்து சரியில்லைனா we have to discard the data. We should not take the data as such. So instrument validation, equipment records should include என்னென்னலாம் இருக்கும், name of the equipment and manufacturer, model or type for the identification, serial number, date equipment was received in the laboratory, copy of manufacturer's operating instruction. இதெல்லாம் இருக்கணும் ஒரு instrumentலயுமே. This has to be there. So if you are visiting a laboratory, in the instrument particular, if you are having hot air oven, in any of the site, the label will be pasted. What is the name of the equipment? Who is the manufacturer? What is the model of the equipment? For the any serial number is available with the model, and then when it is received, and then if it is there, the operating procedure will be separately maintained. So this is the general equipment record. Okay, easy understood பண்றதுக்கு this will be labeled in the equipment itself. The manufacturing protocol motto, it will be separately maintained. So reagent materials certification. So next important thing is reagents or the material certification. So this policy should assure, this policy is to assure that reagents used are specified in the standard operating procedure. So expiry on a reagents we should not use it. So it should also compile with the SOP procedure. Purchasing and testing should be handled by a quality assurance program. So purchase of the reagent or the material and testing. So purchase ஆகி வந்துச்சுன்னா அதோட அது நிக்க மாட்டாங்க. The quality assurance department will take the samples in the pharmaceutical industry, for example in building. So இப்போ வந்து ஒரு reagent or the material is coming inside our industry. So the sample will be taken and it will be analyzed whether it is complies with the results complies with the standard operating procedure. அவங்க கொடுத்துக்கிற லிமிட்ஸ் படி அந்த ரிசல்ட்ஸ் வருதான்னு டெஸ்ட் பண்ணுவாங்க. Whether it is expired or it is in a current condition, whether it is giving maximum efficiency, இதெல்லாம் டெஸ்ட் பண்ணிட்டு தான் and then the material will go on to the research team or in the production team. ஓகேவா, அது இல்லைன்னா அதை அப்படியே டிஸ்கார்ட் பண்ணி they will send it off. So the reagents and solution, என்னென்ன requirements, the reagent and solution should be labeled, properly labeled. Deteriorated and outdated reagents and solutions shall not be used. So deteriorate ஆயிடுச்சு, கெட்டு போயிடுச்சு, சரியில்லை, இல்லை outdated expired reagents we should not use. Include date opened, நம்ம எப்போ open பண்ணோமோ we have to mention the date. For each and every reagents and the solutions we have to mention the date on which it was opened. ஒரு சிலது open பண்ணி we can use it for a prolonged time, but some reagents it should be used within the prescribed limit time. Stored under ambient temperature. Fridge-ல வைக்கிறது கொண்டு போய் we can't keep it under room temperature. Room temperature-ல இருக்கிறது கொண்டு போய் we can't keep it under fridge refrigerated conditions. So storage under the ambient temperature and expiration date is needed. It is most important. So analyst certification. So some acceptable proof of satisfactory training and or competence with the specific laboratory procedures must be established for each analyst. Analyst is nothing but who is performing the experiment. Qualification can come from education, experience, or additional trainings, but it should be documented. ஸோ அங்கே இருக்கிற அனலிஸ்ட் வந்து அவங்களோட எஜுகேஷன் ப்ரொஃபைல் வச்சு we can give the job. ஓகேவா? பட் additional trainings must be there for if you are handling particularly with the HPLC, specific training must be needed. If you are handling in gas chromatography, particular training is needed. ஓகேவா? ஸோ அவங்களோட எஜுகேஷனும் எக்ஸ்பீரியன்ஸையும் வச்சு we can give the job profile job, but later on they should have some certification based on the instruments handling. Sufficient people must be there, and requirements of certification will vary according to their needed, according to the equipments they are handling or the research projects. So laboratory certification is needed. It is normally done by an external agency. So நம்ம laboratory வந்து certify பண்ணனும்னா some agency will be there. We have to call those agencies. We have to consolidate our reports. We have to submit it. They will go through our standard and they will give you a certification. இப்போ ISO certification வாங்குறாங்க இல்லையா some laboratories-க்கு அது மாதிரி தான். Evaluation is concerned with issues such as adequate space இருக்கான்னு பார்ப்பாங்க. Adequate space must be there. Ventilation must be there. Storage must be there. It should be hygienic. The environment must be hygienic. So for a laboratory certifications, these are the things needed. So next is specimen sample tracking. So it varies among the laboratories. Okay, it must contain the unmistakable connection between a set of analytical data and specimen or the samples from which they were obtained. So ஒரு சாம்பிள் எடுக்கிறோம், அதை வந்து ட்ராக் பண்ணி we should end it with the report. ஒரு சாம்பிள் எடுக்கிறோம், அதுக்கு we should label it and then we should undergo some studies. After that it has to be converted into another form of formulation, any drug formulation or some other. So அந்த சாம்பிள் ட்ராக்கிங் must be clear. அது வந்து கிளியரா இருக்கணும், அதுல எதுவும் மிஸ்டேக்ஸ் வரக்கூடாது. ஓகேவா? Original source of specimens or samples must be recorded and unmistakably connected with the set of analytical data. அது எதுவும் கரண்ட் டேட்டா கூட மிக்ஸ் ஆகிடக்கூடாது. ஸோ த ஸ்பெசிமன் ஆர் தி சாம்பிள் மஸ்ட் பி ரெக்கார்டட் இன் எ கரெக்ட் ஃபார்ம். ஓகேவா? அப்படி பண்ணாதான் த எண்ட் ரிப்போர்ட் வில் பி கிளியர். So with this clear, did you have any doubts? Anyone have doubts?